Hi, everyone. Thank you for coming. Now that we've heard a little bit of background from Dr. Quick, I'd like to tell you a little bit about what we do at Scintillation Nanotechnologies. Slides are not advancing. So, as we heard from Professor Quick, a lot of the currently available particles for scintillation proximity assays are quite large. They're between 5 and 10 microns in diameter. And so while we were working on an entirely separate project, we thought about what the advantages of decreasing that size of those particles could be and having a controlled surface chemistry. And so our particles are about 300 nanometers in diameter. They're much, much smaller, but they're still scintillating particles. So they have enhanced dispersibility, especially in aqueous samples. So that helps make measurements over time more easily. You don't have to worry about shaking your sample quite as often. And that can also help with improved surface area to volume ratio because our particles are so small. So per milligram, we have a lot more space on our particles than larger microparticles. And because we can control our surface chemistry, our surface of our particles is very easily controlled. And we have yet to find the limits of what we can attach to the surface of our particles. So we have three basic products, and there are three separate ideas behind them. The first is our aquascint particles, which is just a scintillating particle. That could be for very basic detection of low energy radioisotopes. And so we have found that they respond to the low energy radioisotopes like tritium and S35, P33. We've also tested P32, which I couldn't show here. Shortly, we will be testing carbon-14. And we have tested some alpha emitters, which I have not tested here as well. So for just general detection of radioisotopes, aquascint can detect those in a typical liquid scintillation counter. You can also recover them. So even though they are small, you can centrifuge the aquascint particles and recover them. So you can recover your sample and potentially reuse the particles. I don't know if that would really be as useful, but recovering a sample could be very useful in some applications. So we typically do this in those very common Eppendorf benchtop centrifuges, where you can centrifuge 50 milliliter tubes or 50 milliliter tubes for less than 10 minutes. So it doesn't take very long, and you can make a little pellet and get your sample back. If you need specificity, as Professor Quick was mentioning before, we also make scintillation proximity assays. And so we can functionalize the surface of those nanoparticles to make proximity assays. And we've done this in several models to demonstrate that the proximity assay could work. We first just chemically linked tritiated acetic acid actually to the surface of our particles. So this was a direct chemical linking. And you can see the difference between unlinked tritiated acetic acid and linked tritiated acetic acid. So you can see when it's linked and when it's not linked, and it's dependent on concentration. We've also been able to do this by radiolabeling proteins. So we actually, in very inefficiently labeled some nutravidin, but it did become labeled, and we made some biotinylated particles and were able to monitor the binding of this radiolabeled nutravidin to the outside of our nanoSPA particles. Down here, you can see the non-proximity effects and the non-specific absorption of the nutravidin. So some of the nutravidin could stick to the particle and yield a signal, right? But it's much, much less than you see for the signal for specific binding. And while we don't offer this as a product, this is a further demonstration of what can be done with these particles. We were also able to link single-stranded DNA to the outside of our particles and then hybridize a complementary strand to the outside. So we could hybridize a complementary strand that was radiolabeled and see that bind to our particles. This here again, sorry, the red squares are the non-specific absorption down at the bottom. And then we could come in later with a unlabeled complementary strand or a mismatched strand that was different from the complementary strand by, I think it was 4 bases, and then a random strand. And we could move the the tritiated complementary strand off the particles and watch that happen too in a competitive binding assay. So our particles can offer several advantages and are very easily adapted to bind. Like I said, we haven't found the limit of what we can attach to the outside yet. Because of that versatile surface, we can have nanoSPA for a range of applications. I showed you chemical linking, which probably most people would not be necessarily interested in doing, but for other binding assays, for example, for histag proteins, we are able to attach a nickel chelating group to the outside. So you can attach your histag protein to the outside of the nanoSPA particles and do a radioassay that way. We've been able to add streptavidin or biotin to the outside of our particles. In this example on the left, we have streptavidin functionalized particles that we then treated with tritiated biotin. And again, you can see the tritiated biotin binding the streptavidin particle. Down here is the non-specific adsorption of biotin to the particles. We're also making protein A and protein G particles for other applications. So these are readily available for anybody who needs to use them at this point. Our third branch of products is Aquasyn IC for intracellular measurements. This is something that we are getting into now. And that is that because our particles are so small, they will actually go inside of cells. So on the left here, you can see some cells. These are actually CHO cells without any of our particles. And on the right, the particles are shown in green. So these cells have taken in the particles. And the idea is that this could be very useful for uptake assays, for example. If you're watching the uptake of a drug, perhaps in living cells, then you can monitor this over time by putting your scintillator inside of the cells. And I should point out, our particles are not actually green. This is false colored. These particles in this case were red, so they emit at red wavelengths. The viability of the cells after taking in the nanoparticles depends on the cell line. In the cell line that we saw on the previous slide, they tolerate it reasonably well. You can put in 10 micrograms per milliliter for up to 18 hours and still have high cell viability, and that is a fair number of particles. We're working on finding out exactly how many particles that is so that you can correlate uptake of the particles with what your signal really is and have a quantitative value available. But we know that you can actually put in a number of particles without destroying your cells, that they tolerate it pretty well. And this is a very early experiment that I would like to share with you, but we've been able to measure the uptake of tritiated glucose as well. So in those cells that I showed a picture of previously, we incubated them with our Aquasyn IC particles, rinsed away any remaining Aquasyn IC, and then treated those cells with tritiated glucose for a certain amount of time, and then removed them from the surface and actually did some scintillation counting to see if we could determine how much glucose they took in over time. So for cells without any tritium, the signal is quite low. For cells that had tritium or tritiated glucose for only a minute, the signal is still quite low. That was only 1 minute that they had the glucose to take up. After 4 hours, the signal increases, and in 24 hours, it increases further. So this is a work in progress where we have some other experiments that we would like to do to support this. But at this point, we're quite certain that most cell lines will take up our Aquasyn IC, and you can make some measurements. So to summarize, we have Aquasyn, which is just for general detection. There's no functionality on the outside. If you have low energy radioisotopes or other isotopes that are perhaps precious and you would like to recover, it's easily centrifuged and removed from your sample. nanoSPA has added selectivity for proximity assays. And so we have nanoSPA for histags, we have streptavidin functionalized nanoSPA and biotin functionalized nanoSPA, as well as protein A and protein G. And we would be more than happy to hear about anybody's needs in terms of what other kinds of nanoSPA would be most useful for our customers. And we also have Aquasyn, which is for intracellular measurements. So thank you very much. And I'm excited to hear from Professor Coleman to see how he has been using nanoSPA for his tags and how that has been working out for him.